Hi, this is Wang from the Feinstein Institute for Medical Research. Today, I'm going to share with you our journey in the search of late-acting mediators of lethal sepsis that include HMGB1 and procalcitonin or PCTSL. In the lab, experimental sepsis can be simulated either by endotoxemia through the injection of known amount of bacterial endotoxin or lipopolysaccharide or LPS. It can also be induced by a surgical procedure called second ligation and puncture or CLP, which is the most clinically relevant model for human sepsis. In response to lethal endotoxemia or CRP sepsis, animals accumulate early cytokines such as TNF within a few hours in the circulation, and yet most animals die several days later, suggesting that other late-acting mediators may also contribute to the pathogenesis of lethal sepsis. To search for potential late-acting mediators of lethal sepsis, we stimulated macrophage cultures with early cytokines such as TNF, looking for proteins that were released relatively late. A 30-kD protein that was induced by TNF was identified as HMGB1, which is the nuclear protein involved in DNA binding and regulation of gene expressions. This immunostaining of macrophage cultures with HMGB1 specific antibodies, showing the nuclear localization of HMGB1 in control cells and both nuclear and cytoplasmic localization of HMGB1 in LPS-stimulated macrophages. It suggests that nuclear HMGB1 is actively translocated to the cytosol prior to its subsequent release. In animal model of lethal endotoxemia, HMGB1 is accumulated in the circulation in a delayed fashion, distinguishing itself from early cytokines such as TNF. In animal model of lethal endotoxemia, HMGB1 specific antibodies promoted a dose-dependent protection, supporting a pathogenic role of HMGB1 in lethal systematic inflammation disease. Therefore, in contrast to early cytokines, HMGB1 accumulates in the circulation in a delayed fashion and serves as a late mediator of lethal sepsis. Upon its extracellular release, HMGB1 can bind various pattern recognition receptors and contributes to the pathogenesis of lethal sepsis. On one hand, HMGB1 can bind to TLR4 to induce dysregulated inflammation. On the other hand, it can also bind to RAGE and induce periapoptosis and immunosuppression. Recently, we discovered that a long-derived protein called tetranectin was involved in HMGB1-mediated periapoptosis and immunosuppression. Here shows the identification of tetranectin as a serum protein depleted in septic patients by SDS page gel electrophoresis and mass spec analysis. Tetranectin can be found in the blood of normal healthy controls at fairly high levels. However, its level was markedly reduced in patients with sepsis or septic shock. Similarly, blood level of tetranectin was also time-dependently decreased in animal model of lethal endotoxemia as well as CRP sepsis. Tetranectin was first discovered in 1986 as a plasminogen binding protein. It shares an almost 90% sequence similarity between human and murine counterparts. Tetranectin is expressed in various organs with highest abundance in the lungs. To understand the role for tetranectin in sepsis, we first employed a genetic approach. Genetic depletion of tetranectin rendered mice more susceptible to lethal sepsis, suggesting a protective role for tetranectin in lethal systematic inflammation. We then further tested its role in sepsis using pharmacological strategies. Specifically, the supplementation of tetranectin at subphysiological doses conferred protection against lethal endotoxemia and sepsis, confirming a protective role of tetranectin in lethal systematic inflammation. We then generated polyclonal antibodies against recombinant tetranectin and further examined their effect on the outcome of lethal sepsis. Surprisingly, we saw a rather divergent effect of different antibodies on the outcome of lethal sepsis. Some antibodies are protective and others are harmful. The characterization of the epitope profile by dotting blotting analysis identified a unique epitope, the P5, for these protective polyclonal antibodies. We then generated monoclonal antibodies against human and murine tetranectin and particularly screened for the clones that are reactive with the P5 epitope. We obtained two panels of monoclonal antibodies that recognize two different epitopes, the P2 and P5. In contrast to the P2 reactive monoclonal antibodies, which did not affect animal survival, the P5 reactive monoclonal antibodies rescued mice from lethal sepsis even when the first dose was given at 24 hours post-CRP. To understand how these P5 reactive monoclonal antibodies confer protection against sepsis, we tested the possibility of tetranectin HMDB1 interaction. SPR analysis revealed a strong interaction between these two proteins, suggesting that tetranectin and HMDB1 may form complexes which can then be engulfed by macrophages through endocytosis. Indeed, when tetranectin and fluorescent-labeled HMDB1 was added together to macrophage cultures, tetranectin markedly enhanced the cellular uptake of the fluorescent-labeled HMDB1. When fluorescent-labeled tetranectin and HMDB1 was added concurrently to macrophage cultures, it showed a dramatic co-localization in the cytosol, confirming that tetranectin HMD1 complexes are engulfed by macrophages, possibly through endocytosis. As a possible consequence, the co-addition of tetranectin and HMD1 synergistically increased macrophage cell death, as judged by the trypan blue exclusion and LDH release. Consistently, the co-addition of tetranectin and HMDB1 induced the formation of ASC specs, which are markers of para-apoptosis of human macrophages. In animal model of CRP sepsis, the P5 reactive protective monoclonal antibodies effectively prevented sepsis-induced tetronictin depletion. To understand how these P5 reactive antibodies prevented sepsis-induced tetronictin depletion, we examined their effect on tetronictin-HMDB1 interactions. SPR analysis revealed that these P5 reactive antibodies effectively disrupted tetronictin and HMD1 interaction, as indicated by the almost 10 to 50-fold increase in the KD for tetronictin and HMD1 interaction. And now we switch the gear to the second part of the talk, the identification of pro-caspase-L or PCTSL as an inducible late-acting mediator of lethal sepsis. And this slide shows identification of PCTSL as an assay-inducible protein in macrophage cultures. And this slide shows the definition of pre-caspase-L, pro-caspase-L, and caspase-L. In response to bacterial endotoxin LPS or human serum amyloid A or SA, human PBMCs upregulate and secrete PCT cell in a time and dose-dependent manner. In experimental sepsis, the CTSL mRNA levels was upregulated in many tissues at 24 hours post-CRP. Consistently, blood PCTSL levels was elevated in delayed fashion. In the blood of normal healthy controls, PCTSL was not detected. However, in patients with sepsis or septic shock, blood PCTSL levels were markedly elevated and parallels with the appearance of few surrogate markers of sepsis, such as interleukin 6, interleukin 8, MCP1, and GRO. Blood PCTSL levels also positively correlated with the clinical score as well as the level of a few surrogate markers of sepsis. To understand the biological consequence of PCTSL release, we tested its possibility to interact with pattern recognition receptors. SPR analysis revealed a strong binding of PCTSL with TLR4 and RAGE. We then examined the impact of PCTSL on gene expression in experimental settings. The intraperitoneal administration for PCTSL upregulated the expression of many genes, including fibrinogen alpha, beta, and gamma in the liver. Consistently, administration of PCTSL induced marked thrombosis in the liver of wild-type animals, but not much so in mutant mice defective in TLR4 and RAGE. It suggests that PCTSL induced liver injury through both TLR4 and RAGE-dependent mechanisms. In contrast, genetic disruption of CTSL expression rendered the mice more resistant to sepsis-induced liver injury, confirming a pathogenic role of PCTSL in lethal sepsis. We then generated monoclonal antibodies against two specific epitopes, the P12 and P13 epitope of PCTSL. In contrast to the P12 reactive antibodies, which did not affect sepsis survival, the P13 reactive monoclonal antibodies uniformly rescued mice from lethal sepsis even when the first dose was given at 24 hours post-CRP, confirming that pCTSL serves as a late-acting pathogenic mediator of lethal sepsis. The monoclonal antibody-mediated protection was associated with a significant attenuation of sepsis-induced accumulation of several surrogate markers of sepsis that include interleukin-6, MIP2, and KC. In vitro, these protective anti-pCTLs monoclonal antibodies also dramatically reduced pCTLs-induced production of cytokines and chemokines in human PBMCs. Mechanistically, these pCTSL neutralizing antibodies effectively prevented pCTSL infection with both TLR4 and RAGE, as indicated by the almost 50 and 10-fold increase in the KD for pCTSL infection with TLR4 and RAGE respectively. This is a class pro docking of pCTSL infection with TLR4 and RAGE, showing the relative localization of the p13 epitope for the protective antibodies on the pCTSL protein. To develop potential therapeutic strategies for sepsis, we screened a library of 800 natural products for pCTS-specific inhibitors, and this effort has led to the identification of lipophilic molecule, lanosterol, as a selective inhibitor of pCTSL-induced TNF production in macrophage cultures. When dissolved in organic solvents such as ethanol, lanosterol selectively inhibited pCTSL-induced production of several cytokines and chemokines in human PBMCs. To increase its bioavailability, we generated phospholipid DPPC-based liposome nanoparticles that carries either lanosterol or cholesterol as a control, and tested their impact on pCTSL-induced inflammation both in vitro and in vivo. In vitro, the lanosterol-carrying liposomes effectively inhibited pCTSL-induced production of several chemokines, confirming the pCTSL inhibiting properties of lanosterol. In animal model for CRP sepsis, the lanosterol-carrying liposome nanoparticles effectively rescued mice from lethal sepsis even when the first dose was given at 24 hours post-CRP. This protection was associated with the parallel inhibition of sepsis-induced liver injury. Similarly, the protection was also associated with the parallel inhibition of sepsis-induced systematic accumulation of several cytokines and chemokines. In summary, in response to microbial infections, pathogen-associated molecular pathogens stimulate innate immune cells to sequentially release early and intermediate cytokines. The early and intermediate cytokine then triggers the release of later acting mediators such as HMDB1 and pCTSL, which collectively triggers both dysregulated inflammation and immunosuppression that collectively contribute to the pathogenesis of lethal sepsis. Our research has been supported by the National Institute of General Medical Science, as well as the National Center of Complementary and Alternative Medicine. Finally, We would like to thank all the members in my lab, as well as our close collaborators, both in the US and elsewhere. Thank you for your attention.